In this topic, we shall look at firstly the mode of action of enzymes, what they do and how they do it, and secondly, the properties of enzymes. This is related to their tertiary structure, and we shall see how changes in pH and temperature and the presence of inhibitors affects their properties and action. So, let's look at their mode of action. Enzymes are biological catalysts. They lower the activation energy of reactions so that the metabolic reactions that take place in cells can do so at a relatively low temperature that will not denature other proteins, such as membrane channel proteins, and will also proceed quickly enough to sustain life. These metabolic reactions would take place without enzymes, but too slowly to sustain life. A meal would take about 50 years to digest. So enzymes speed up biological and metabolic reactions. They are usually proteins, although some molecules or RNA can act as enzymes. These are called ribozymes. The reactions in cells, like all chemical reactions, need some activation energy to get them going. It is like rolling a ball up the hill so that it can run down on the other side. The presence of an enzyme lowers the amount of activation energy needed to get a metabolic reaction going. All molecules possess kinetic energy and are constantly in random motion. As they move around, they may bump into each other or into substrate molecules. If they collide with substrate molecules, then the substrate molecules will fit into the active site and be brought in close proximity to one another. The enzyme molecules and substrate molecules form enzyme-substrate complexes, where the substrate molecules are held in the cleft of the active site by hydrogen bonds. In anabolic reactions, when the substrate molecules are brought together, a bond can form between them to produce a larger product molecule. For example, two molecules of glucose can react together to form a molecule of maltose. In catabolic reactions, a large substrate molecule may have a bond in it broken or hydrolyzed to form smaller product molecules. For example, a molecule of sucrose can be hydrolyzed to form a molecule of glucose and a molecule of fructose. At the end of the reactions, the product molecules are released and the enzyme molecule is free to combine with another substrate molecule to form another enzyme-substrate complex. This means that a small amount of enzyme can convert a large amount of substrate. The number of enzyme-substrate complexes per minute that can be formed by one enzyme molecule is its turnover number. This may be as high as 6 million for some enzyme-catalyzed reactions. Let's now look at the properties of enzymes. We'll start by looking at the effect of changing the temperature on the rate of an enzyme-catalyzed reaction. At low temperatures, molecules have smaller amounts of kinetic energy, and so there will be fewer collisions between enzyme and substrate molecules. Fewer enzyme-substrate complexes formed and a lower rate of reaction. As the temperature increases, the molecules gain more kinetic energy and so more collisions occur and the rate of reaction increases. However, if the temperature becomes too high, then the enzyme molecules vibrate and bonds holding the tertiary structure in place are disrupted. This means that the shape of the active site changes and is no longer complementary to the shape of the substrate molecule, so the rate of reaction slows. When the shape of the active site is fully unraveled and the substrate molecule will not fit at all, then the enzyme is said to be denatured and there is no reaction. The temperature at which the rate of reaction is fastest is the enzyme's optimum temperature. Some enzymes have a very low optimum temperature. Some have a very high optimum temperature. Some bacteria live in thermal oceanic vents or hot springs, and their enzymes function at high temperatures. Some bacteria live in very hot sulfur springs. In addition to tolerating very hot temperatures, they, and therefore their enzymes, are also tolerating very low pH values. Now, let's look at the effect of changing pH on enzyme action. Enzymes operate within narrow limits of pH. This means that they have an optimum pH at which their rate of reaction is fastest, and they will only function within a small range of pH either side of the optimum. Changes of pH disrupt the hydrogen bonds within the enzyme molecules and so will alter the shape of the active site. This means that the substrate molecules will not fit into the active site so well and the rate of reaction will slow. At extremes of pH, the enzyme molecules will be denatured. Some enzymes work best at pH 7, neutral, some at high pH, alkaline, and some at low pH, acid. There are enzymes in blood and it is therefore very important that the pH of the blood is maintained within narrow limits at around 7.2. Hemoglobin and other proteins, as well as certain other chemicals in the blood, act as buffers. They can either soak up or release hydrogen ions to resist changes of pH. The rate of reaction of an enzyme-controlled reaction can be lowered by the presence of enzyme inhibitors. Metabolism takes place in a series of reactions, each catalyzed by a different enzyme. When the concentration of end product increases, it will inhibit one of the enzymes involved in the metabolic pathway so that the production of an end product ceases. As the concentration of end product reduces, then its inhibitory effect is removed and the pathway operates again. Some enzyme inhibitors have a molecular shape very similar to that shape of the substrate molecule. It may therefore bind briefly to the enzyme's active site and prevent the substrate molecule from binding and so reduce the rate of the enzyme catalyzed reaction. The inhibitor molecule does not bind permanently to the active site. If the concentration of substrate is increased, there will be more substrate molecules. This means that the chances of enzyme molecules colliding with substrate molecules will increase and the rate of reaction will increase again. The inhibitor molecules and substrate molecules are competing for the enzyme's active site, but this type of inhibition is not permanent and so is reversible. If the inhibitor binds permanently to the active site of the enzyme, then the inhibition is non-reversible and is therefore non-competitive as, no matter how much more the concentration of the substrate is increased, the rate of reaction will not speed up again. If the inhibitor binds to another part of the enzyme, an allosteric site, then it may cause the active site to change shape so that the substrate molecule no longer fits. This is non-competitive as the inhibitor is not competing with the substrate for the active site. It may also be called allosteric inhibition. It may be reversible if the inhibitor binds to the allosteric site only briefly, or irreversible if the inhibitor binds to the allosteric site permanently. Digitalis is extracted from purple foxglove leaves and contains digitoxin and digitalin. Digitalin is a non-competitive inhibitor. It has been used for centuries to treat congestive heart failure. It binds to a part of the sodium-potassium pump's ATPase enzyme and so leads to a buildup of sodium ions inside the heart muscle cells. This stimulates the sodium-calcium antiport channels in the membrane that pump sodium ions out and calcium ions in. The influx of calcium ions increase the force of heart muscle contraction.